The topic I've chosen to research is the effect of natural cleaning products on bacterial growth on the anterior surface of mobile phones. Now, I'll tell you a little bit about me. My name is Colten Adkins, and I currently attend Anterior High School. I'll be attending the College of William & Mary in the fall, while I'll be majoring in chemistry. As for why I chose this project, I have three main reasons. The first being my personal interest in mobile phone cleanliness. As an owner mobile myself, I wanted to learn more about how to keep my device clean and free from bacteria as I continue my research into this topic. This leads to my second reason, which is because of lack of research done on natural disinfectants, even mobile phone cleaners as a whole. I was not able to learn more about many types of disinfectants that are typically used on mobile phones, as the research done is sparse. My third and final reason for choosing this project is because of the benefit of using environmentally friendly disinfectants on mobile phones. This will allow for people to use these products without having to worry about how they're harming the world around them. Before I conducted my experiment, I researched several studies to strengthen my understanding of this topic. Through this research, I found that more often than not, mobile phones tend to be highly contaminated with bacteria. There are many types of bacteria typically found in mobile phones, but the most common variant that appears is Coagulus staphylococcus, which is most commonly found in human skin. While it's not typically dangerous, it has the possibility of causing serious medical issues, even death. Even though the danger to the bacteria on these devices can be immense, previous research has shown that a large number of people do not even know that these devices are so heavily contaminated. In addition to bacterial growth, my research of previous studies led me to find that disinfectants are typically very effective at reducing mobile phone contamination. Even so, there's a very limited amount of research done on how these products affect mobile phone contamination, and even less done on a variety of products. The majority of research that has been done on these products tests the effectiveness of 70% isopropyl alcohol. Because of this, there is little to no research completed on products such as natural disinfectants. While furthering my understanding of this topic, I also found that methodologies, particularly the temperature of agar and petri dishes, tend to be very similar among most studies within this field. This is most likely due to the majority of research being done almost identically, with only some variation in instances of location or equipment used. While factors and methodology are typically the same for studies in this field, the amount of time which petri dishes are incubated for often differs from one state to the next. This small difference is problematic, as it can be what's causing different results between studies. There are also problems regarding which type of mobile phones swap for testing when gathering samples. Studies that indicate which type of mobile phone they swap for testing tend to do so without reasoning, making it seem as if the decision was arbitrarily made. Studies that indicate which type of mobile phone they swap for testing. Determining which type of mobile phone swap when gathering samples is an important part of the methodology, so it's important to provide some reasoning as to which side or sides are being swapped. Because the anterior, otherwise known as the front, surface of mobile phones come into contact with fingers whenever the screen is touched, and device owners often touch the front of the screen when they make calls, I decided to test the anterior surface in my research. I will now provide my reasoning for why this research needs to be conducted. Even though there are some studies that test the effectiveness of cleaning products on mobile phones, the cleaners that use alternative products, such as the aforementioned 70% isopropyl alcohol. In addition, there was research in the past that tested the effectiveness of natural cleaners, but because it was done on the surface of household surfaces, the results were found are not applicable to mobile phones. Taking all this into consideration, this research needs to be conducted in order to fill these gaps for alternative products or surfaces for use. This gap led me to my research question: how do natural cleaning products affect bacterial growth on the anterior surface of mobile phones? The research hypothesis I formed in relation to this question was that if the misfired screen cleaners used to disinfect mobile phones, then the bacteria reduction will be greater than the other products. As the products that I chose to use in my experiment had not been previously tested, I did my own research into them based on online reviews and concluded that the misfired disinfectant is most likely to be the most effective. I then formed my null hypothesis as a means to state that there is no statistical difference between variables that I tested, and it reads that if the misfired screen cleaners used to disinfect mobile phones, then there will be no difference in bacteria reduction. The following are the variables that have been identified for my experiment. The independent variable is the different types of disinfectants. The dependent variable is the growth of bacteria. The control group that was used in my experiment was tested using no products. And the constants are the temperature at which petri dishes were incubated, the agar used in those petri dishes, being nutrient agar, and a microfiber cloth used to apply the products. As for the main materials used to conduct my experiment, I gathered the following disinfectant products: Mystify, Better Life, Pathosands, and 7th generation natural screen cleaners. In addition, I also gathered the mobile phones for my participants and a microfiber cloth in order to apply the products. Moving on, the sample size of my experiment was 64. 32 were from my control, and the rest consisted of 8 from each type of disinfectant group. In addition, the sample population for my study was all bacteria, as there was no one specific type that I tested for. To begin my methodology, I obtained consent from administration, parents, and participants prior to gathering all my materials. I then got my participants to sign on a sheet of paper before I collected their mobile devices. After this, I dipped sterile swabs into distilled water, then swabbed the phones before applying any products in order to gather my control sample. The swabbing of phones can be seen in the picture on the left. These swabs are placed in labeled Ziploc bags in order to be later transported to the lab. I then applied one of the 4 natural products using a microfiber cloth, which can be seen in the picture on the right. The mobile phones were then swabbed again using a sterile swab into distilled water in order to collect my treatment sample. Once again, these swabs were placed in labeled Ziploc bags. After gathering all my swabs, participants then signed out on the same aforementioned sheet of paper. Then all the swabs were taken and inoculated onto petri dishes. Following the inoculation of petri dishes, which can be seen in the picture on the left, they would take shut and incubate for 48 hours at 37 degrees Celsius before I counted the number of bacterial colonies. The picture on the right shows me counting colonies. The number of colonies found before and after application of products were then recorded onto an index card. Steps 2 through 9 of my methodology were then repeated until all different products had been tested. After collecting all my data, participants were debriefed, and finally I compiled all my data into a spreadsheet and conducted statistical analysis. Here I created a graph that provides a great representation for the change in growth of bacteria after the products were applied. To start with, both the Pathosands and 7th generation groups had outliers. These outliers will show a large increase in bacterial growth following the application of these products, suggesting that when used, they may cause an increase in bacterial growth without any new piece. Another thing that stood out about this graph is that the misfied group had a small standard deviation, or spread of data points, out of all the groups. This may suggest that the misfied disinfectant is most likely to have similar results when used multiple times with mobile phones. The Pathosands and 7th generation groups would have had even lower standard deviations if not for the outliers in the groups. Therefore, the same conclusion could be made, but with less confidence that it's likely to be true. Finally, the Better Life group had the lowest mean, suggesting that it's the most effective of the tested disinfectants at reducing mobile phone contamination. After completing my experiment, I performed a one-way ANOVA test on the growth of bacteria to determine the p-value of my groups. From it, I was able to determine that with a p-value of 0.48, there is not sufficient evidence at the 0.05 significance level to conclude that the misfied disinfectant had the greatest effect at reducing mobile phone contamination. Even though my research hypothesis was not supported, I learned that the misfied disinfectant, along with the other disinfectants, can have varying effects at reducing mobile phone contamination. In conclusion, natural disinfectants could be used to reduce the amount of bacterial mobile phones, but the increase in bacterial growth that may occur, such as the 2 outliers I mentioned previously, could make it not worth using them. Either way, I believe it is up to device owners themselves to make this decision. But regardless, my findings suggest that these products should not be used in order to reduce mobile phone contamination. The majority of previous research regarding the contamination of mobile phones found that there's a decent amount of mobile phones that are not contaminated at all with bacteria. This is not matched with my findings, as all the mobile phones that I tested were at least somewhat contaminated. The difference is likely due to the fact that most previous research was done using the mobile phones of healthcare workers, whereas the participants in my study consisted of teachers and students. In addition, previous researchers found that natural disinfectants were less effective than the average cleaner at reducing bacteria. This research supports my findings, but as the testing was not completed on mobile phones, the findings are not completely applicable. Regardless, the similarities are still important to note, as it suggests that it may be better to use disinfectants that are not natural. There were many limitations within my study that future researchers should attempt to improve upon. To begin with, a convenient sample is used to obtain participants in my study. If a convenient sample had not been used, the results gathered would have more likely been representative of the population as a whole. Another limitation regarding my sample was the size of it. If a larger sample size had been used, the results gathered would have likely been more accurate. Therefore, future research should attempt to use a larger sample size. An additional problem that I faced while conducting my experiment was that clear instructions for how to apply the product was only given for the misapplied disinfectant. Because of this, I had to use these instructions to apply all of the products. If the proper method of applying each of the products had been used, the results would have likely been effective. Other than only using products that list clear instructions on how to apply them, there's not really much that future researchers could do to solve this problem. The final limitation of my study is that whether or not mold fins contain a screen protector prior to data collection was not determined. This is problematic because it may have affected how bacteria grows on mold fins as well as how well disinfectants work on mold fins. Therefore, providing variables such as this one should be considered more in the future. To continue off of my previous mentioning of screen protectors, I believe that this provides for a great idea for future researchers to experiment with. For example, researchers could test if screen protectors affect how mold fin contamination is affected, as well as if disinfectants affect how well, if screen protectors affect how well disinfectants work on mold fins. In addition, future research should test the different cleaners from the ones that are used in this study. There are many different types of disinfectants with different ingredients, so it serves to reason that the four that I tested do not represent how likely all disinfectants are likely to be. In addition to learning new information about my research topic, I also learned a lot about how the research process as a whole works. Completing this research has allowed me to gain more knowledge about the inquiry process, how to properly manage my time, and how to plan out events ahead of time. Also, this whole process has taught me a lot about how to persevere through difficulties. For example, I had to change my dates for data collection on multiple occasions due to school closings because of weather. I believe this newfound knowledge will continue to benefit me as I head to college and even beyond that. I've also learned just how much I enjoy working in labs. This is especially important because, as I previously mentioned, I'll be majoring in chemistry in college. Therefore, I'll likely be partaking in many more labs in the near future. I thoroughly enjoy this whole process, and I'm glad it allowed me to learn so much about research and myself. To conclude my presentation, I'd like to give special thanks to all those who helped me along this process. First and foremost, I'd like to thank all my participants, as I would not have been able to collect any data without them. The main person who assisted me throughout this entire process was Ms. Long, so I'd also like to give a big thanks to her. In addition, both Ms. Dillon and Ms. Hardy helped me revise my methodology, so I'd like to thank both of them. I would also like to thank Dr. Bocock for giving me permission to stay after and complete my lab work at the Institute. Finally, I would like to thank Dr. Jones and Ms. Bowman for approving my research to begin with. At this time, I'll answer any questions all of them I have. Yes, Ms. Wall.
So I see you used 4 different products as the natural disinfectants, but do you know what the active agent in each of those was that is the disinfectant feature?
So Ms. Walls asked if I knew the active ingredients in the disinfectants that I tested. So that was not within the scope of my research, so no, I do not, sorry. Yes, Mr. Talarico.
So I have a question about the methodology. So you talked about cleaning with the microfiber cloth. Was the same microfiber cloth used for everything, or was it washed between cleanings?
So Mr. Talarico would like to know if the same microfiber cloth was used for each disinfectant, or whether it was cleaned beforehand. So I did use the same one for each time, and no, it was not. That's why I listed it in my limitations, because I believe that it may have affected the results.
And do you think that explains why the outliers were there?
So Ms. Craig asked if it could explain the outliers there. It could, but also the outliers were all from the same mobile phone. Therefore, I believe that it's something to do with the mobile phone specifically, and not the microfiber cloth, but it could have affected something else.
So I have one more follow-up question to that. So as you swabbed the phones for testing, did you swab the phones in the same place, or did you kind of change position?
So Mr. Talarico would like to know if I swabbed the phones in the same place. I did swab them all in the same place. It was all over the interior surface, so all on the screen. I swabbed all around for around 10 seconds.